Hello, my name is Ellefson. I am a material science major and senior at UW-Eau.
And I am Cuyler Monahan, a material science and engineering major at the University of Wisconsin-Eau Claire. Our co-author and collaborator at Mayo Clinic is Dr. Jeremy McBride, and our primary investigator at University of Wisconsin-Eau Claire is Dr. Elizabeth Glogowski. While I start, I want to just mention one thing. Cancer will affect most of our lives. In fact, 2 in 5 people will develop cancer in their lifetime, and you may already know someone who has cancer. And if not, it is likely that someone you know will develop cancer. This is why the work that we are doing is so important. With the new materials that we are developing, we are working to decrease the complications and side effects of tumor ablation. During ablation, doctors use radio frequencies, burning and freezing to destroy tumorous cells. Some of the drawbacks of ablation include the fact that surrounding tissue can be harmed due to the lack of insulation on the target organ. Insulating the organ from the surroundings is very important, and isolating the organ is also an important thing to control. Current methods to do this include injecting water or CO2 into the body to insulate the organ from the surrounding tissue. Some issues with this include the fact that water can flow away from the tumor site and won't maintain contact with the organ. This leads to a need for a greater volume during surgery. Gas cannot be controlled as easily and can leave the cavity. To solve all of these problems, our research has been focused on developing an injectable foam. Our primary focus was to create a foam that remains in the tumor site and maintain contact with the target organ. We were trying to develop and improve the mechanical properties of this foam so that it can maintain contact for at least 1 hour. As you can see on the right side of the screen, our foam that we are developing is on the left, while the standard foam is on the right. You can watch the collapse over time. We use small batches to test different additives and use different methods to test the stability of the foam.
These are the results of our project, which has defined stability as the foam holding up against weight over time. The baseline of the standard solution shows a gradual decrease in volume and therefore stability over the course of an hour. The essential data points are at 30 and 60 minutes, marked with a dashed vertical line. The baseline is shown in red and has a stability of 77% and 4% at the 30 and 60 minute marks respectively, showing almost total collapse. The foam with the primary additive in the solution is marked in blue and has a stability of 93% and 87% respectively. Collapse over time is important because the foam needs to last the duration of the surgery, yet degrade and disperse inside the body after the procedure is complete. The primary additive is over 20 times more stable on average, which shows significant change in the mechanical properties of the foam. The current process of enhancement leads toward a surgical grade foam that has the desired properties to make it a viable candidate during tumor treatment. Our continued research will focus on secondary additives to further increase the stability of the foam and improve patient recovery through greater comfort. We have already started investigating complementary characterization methods seen on the right. That's an image I took of our baseline using a BX51 light microscope at 20 times magnification. It is filtered to become a binary image to show the bubbles inside the matrix of the solution. An area of interest for data collection is the size and special density of the bubbles in the foam. The smaller the bubbles and the more they fill space, higher density, the more stable the foam. Biocompatibility is a crucial aspect for the overall performance of the foam. All materials selected are approved by the FDA. We want to investigate how the foam changes and interacts with bodily conditions such as temperature and enzymes. This time we'd like to acknowledge the Material Science and Engineering Department and Center of UW-Eau Claire for the lab space and equipment that we use. And our project is only possible through the collaborative Mayo Clinic and UW-DC student funding. Thank you very much.